Into science. I'm very biased because I love science. I trained in biology and bioinformatics, and I get to work with Ali, and it's always fascinating to hear about where AI is actually moving in biology. It's probably one of the most exciting domains here. So I figured maybe to start, it's pretty clear, at least to a lot of us in the space, that every other month or something in Nature or Cell or Science, which are like the most popular journals out there, if you have a kind of old school biological analysis method, it's getting just like ripped and replaced with something that was deep learning based, whether it's images or proteins or whatever modality you want. And so maybe like in that context of like so much is happening in biology, what is it that you chose to work on and why?
Yeah, hey everyone. So I think I'll start with a hyper-specific answer to that question, and I'll zoom out afterwards. Profiling myself, we desire to really have full mastery over the human genome with AI, to be able to cure basically any type of disease using AI going forward. And to take a step back from there specifically, we typically build language models based on protein data. To make biology programmable. Now, what does that mean? What are proteins? I'll kind of set the stage of biology as a whole. So proteins are these molecular machines. Think these molecules that do these exquisite functions. They enable us to breathe, they enable us to see. Basically, for any disease application as well, you can point to a protein or constellation of proteins, for example, that are responsible for that specifically. And beyond human healthcare, proteins are responsible for detergents, right? I went to a site that makes the equivalent of Tide Pods, for example, and they wanted us to build proteins for stain removal, for example. Environmental applications, agricultural applications. We have these partnerships with large agricultural companies that have operations all over the globe specifically, and they're doing protein engineering to make yields better to combat climate change, for example. So they're incredibly small, complex machines. And really, at the end of the day, like let's take AI out of the equation altogether, how we've gone about finding these high impact solutions that affect all of our lives in every imaginable way specifically has been through random discovery. By finding random stuff in nature and repurposing it for our applications, right? Like that, Alexander Fleming example of finding antibiotics, for example, the discovery of antibiotics within the petri dish, for example, that's not the exception, that's the rule. CRISPR-Cas9 that won the Nobel Prize, for example, for gene editing purposes. It was actually early discoveries were in a Dannon yogurt, Danisco yogurt facility. The same yogurt bottles you get at Whole Foods or whatever else, right? Scientists were looking at these interesting phenomena that was occurring in bacteria and found these proteins and were like, oh, maybe we can repurpose this and cram it into human therapeutic applications, and maybe that could cure disease, right? Like that's the way we've gone about building biological solutions. Just finding random shit in nature and repurposing it for really hard problems. And what we're trying to do, and this isn't just, the mission is beyond myself, taking off my individual hat, even taking off my profile hat. The mission is much greater than this. It's a societal level mission, where we're trying to move the field away from random discovery into bespoke design, to intentional design, where we have a problem in front of us, right? We have a virus we're trying to neutralize, for example. A crop we're trying to improve the yield of, for example. There's a kid, there's literally, I talk to clinicians, there are kids that have rare diseases that will die in 2 years. Like it's known they will die in 2 years. And they have a mutation in their genome that if you can only flip that mutation, flip that nucleotide specifically, right, back from an unhealthy state to a healthy state, you can give them a chance to actually live. These are concrete examples. This isn't hyperbole. I'm speaking with clinicians for this specifically, right? And if we can have a way, a technique to actually design these molecules from scratch, right, that can enable so much, like across so many different industries.
Yeah.
So yeah,
just be super careful here that you don't fall off.
Okay, yeah.
Just don't give a buzz.
Try to stay alive.
Yeah. And so you did machine learning in grad school. Like, so you're not a biologist by trade. Like, you moved into this, right? I'm a biologist by trade. No. And so I think what I love people to understand is like, what is the intuition that you have, like, as a computer scientist, machine learner, moving into this like fuzzy, domain where we're doing doing random discovery and like squeezing liquids in tubes and finding things in yogurt pots.
Totally. So the connection here is that so much of biology can be represented and is faithfully represented as a sequence, right? Similar to words in a sentence, for example, tokens, for example. You can represent proteins as amino acids that form, that can be strung together, right? Like beads in a string or tokens in a given sequence specifically. And that encodes for the structure of the protein, that encodes for the function of the protein, and we can utilize basically the recipes for success here. If I were to take it more personal, like you mentioned specifically, this was like, if I rewind to 2019, this was like right before GPT-2 came out, for example. If people remember, like pre-2019, everyone was working on encoder-based models, BERT-based models, for example, right? Because you could easily kind of measure, glue or super glue these downstream classification or regression tasks. But very few folks were working on like these generative models or autoregressive language models and so on. We had a similar phenomenon in biology as well, where people were focused on these reading of biology tasks, like AlphaFold, for example, taking a sequence and predicting the structure, taking a sequence and predicting a function. No one is really working on how do we just de novo generate these sequences from scratch that would be functional. So I saw that trend happening in NLP, natural language processing. So that was one. Two, looking at like, again, the sequence representation, right? You could faithfully describe the data as a sequence. You can have to cast it in a weird way to fit it like, for a transformer, for example. Three, access to like a really information-rich source of data, right? Like we need that, right? Like we have Common Crawl, right? We have these other, like we have the litany, the whole corpus of human-generated text, right? From all over the world that we can learn, right? From. There's an effective, like, it's information rich. You can do transfer learning as well, right? Like that similar database we actually have for biology in orders of magnitude more as well, right? So for proteins in particular, I can speak for Profluent. When we first started out, we had about 2 billion proteins. The beginning of this year specifically, we got to 80 billion proteins.
How many proteins are there in the world?
Yeah, near infinite. Yeah, so an example is AlphaFold that won the Nobel Prize was exposed to around 2 billion proteins. So we reached the 80 billion proteins. We're on pace now within 6 to 9 months to have around 150 to 200 billion proteins. In terms of tokens, we're well beyond 10 trillion tokens, for example, that we can train our models on specifically. So we have an incredible amount of data. And if you really think about it, like that kind of makes sense. Like nature is complex, right? We've had millions of years of evolution to generate all these samples. So those recipes for success, right, of, okay, a move toward decoder models, proteins can be represented, a lot of biology can be represented as a sequence. We have rich data sets specifically, right? Like that was the playground, right? The ingredients for success for me to just like, and others as well, to just dive headfirst into it and let's see what happens.
Yeah. And so I think that earlier this year you had a Nature paper around this topic of using the technology described for genome editors. Can you maybe like describe why that was important and how it works?
Yeah, so it was the first demonstration of using AI in a meaningful way to generate a gene editor from scratch that can edit the human genome. We share this in human cells, and we actually decided like, hey, this is really great. We'll just open source it, and we let everyone kind of use it for free, basically. And we had like within 24 hours, we had like thousands of people request it specifically. People were building companies using this as well. It's been insane. And honestly, like one of the reasons why we open sourced it is like we don't have a scarcity mindset at Profluent. We have an abundance mindset. Like there's just so many problems to tackle, and very few people are tackling them. So if we just open source the first one, it's okay. Well, there's tons more massive opportunities in front of us as well. The significance of the paper, specifically that you're citing specifically, is again like showing a demonstration also from a protein design perspective as well, where we started out by training these really large models on lots of data, and we found that okay, it can generate something sensible. And by computational metrics. So the difference between proteins and biology and natural language is you can't read it, right? It's not we don't read the language of DNA or proteins specifically, right? So we have to use these proxies to see if something is useful or not, right? If something's the equivalent of being grammatically correct or answering the question at hand, for example. And the early work that we did this on like single domain proteins. like lysozymes. These are like proteins that degrade the cell wall for antibacterial purposes. And then we made this big step going from there to work on a multi-domain protein, such as a Cas protein, CRISPR-Cas protein. So these are like, think of it as like not just one molecular machine, but like multiple machines that are stuck together. And there's also an RNA complex as well, RNA component as well that forms a complex. So yeah, from a perspective of like the next challenge in protein design, we found that it was like really exciting to see that it works. And we actually did baselines with respect to other baseline techniques in structure as well, structure-based techniques, and we found that it works much better. Language models uniquely were positioned for this. And then I think the promise that we're really excited by is that not only can you generate things de novo from scratch that are functional, but then you can have really control, molecular level control with respect to the exact properties you're interested in. So again, like zeroing in on the 3 billion bases of your human genome specifically, and finding one specific base, right? One adenine, for example, that you want to flip to guanine, for example. Yeah, I think that level of molecular control is what we're seeking and what we're building toward. And I think that's what's exciting everyone.
Yeah. And to what extent would you rate the quality today of these like AI co-scientists? Like, do you use any of them? Have you used this for like, I don't know, market mapping or like finding new targets, or is it going to be like the one-man biotech or one-woman biotech?
It's true, yeah. I think, I mean, I honestly like use it all the time. I think again, because like I'm not a biologist, I'm in a new domain, and there's just so many nooks and crannies of this domain that it's just so fantastic to just like speak with, take morning walks, for example. I actually did a little thought experiment where like I hired an equivalent of McKinsey consultant to answer a question for me, and then I just had GQ Pro do it as well. And I saw the end results, and I was like, okay, I just paid you like hundreds of thousands of dollars. Sorry, I did. And then I compared it to my, what do you call it, like $30 a month subscription. And I was like, I think this was pretty much the same work product at the end of the day. So like, yeah, it's crazy how much is actually influencing real decisions, whether commercial decisions or also just scientific decisions as to exploring and scaffolding a new space that you haven't thought of before. Yeah. So yeah, I'm incredibly bullish of like, AI scientists work as a whole. I think there's real value to it.
Yeah, yeah. And on the last session, I guess we talked about what's like the next step, what's like the recipe for success in model capabilities, and particularly in your domain, I guess like the model is not in and of itself like the entire problem that biopharma has. Like there's a lot of other moving parts here. So maybe two-part question is like, what's like the next step you're doing? What's like Progen 4? And like, what else do you have to do to build like a differentiated biopharma company in what is the data environment where everybody's Debbie Downer?
Totally, yeah. That's the last question, the good one. I feel like if everyone's excited by something, like I get less excited, and if everyone's against something, then I'm more excited, honestly. In no particular order, I think we're really excited about scaling laws, generally speaking, that we're witnessing. And what's different here is that I really think like the distribution of data that you feed into a model is incredibly important. Like that's not a hot take, right? Like if I trained a language model only on like Reddit subthreads, for example, and then wanted it to generate like a Wikipedia-like document, like that's it's the pre-trained distribution matters a lot, right? What's challenging within the biology domain, again, what I mentioned before, is that it's not immediately interpretable because we don't read and write biology, right? So we lack like enough of an understanding, and not only just like enough resources to really understand what's latent, what's within these massive data sets that we have. So I think that's like a huge angle here that we're investing in, and then thinking and trying to extract and enhance out basically like how these models can be really powerful representations that in post-training techniques or alignment-based techniques, for example, you can really see these really fantastic scaling laws. So I think looking at examining what scaling laws exist and what the benefit and utility of data scaling specifically, I think is one frontier that we're really pushing. I think more practically speaking, outside of the AI side of things, like we just want to work with people who have real problems. Like we're excited by the ability for AI to be an accelerant insofar as like it makes things more efficient. It makes something less costly, for example. Your shots on goal, your percentage increases. But what really excites me is working on spaces where you just simply do not have a solution for it. Like I mentioned before, like we talk to clinicians where these children like literally do not have a solution.
Well, there was a case earlier this year of a baby who was cured from a genetic disease, right?
Totally, exactly, yeah. There's these fantastic stories of like, there just was no solution for it before, there was no drug for it before, and if only we can have a technique to truly make personalized medicines on demand, that could just, it goes from not having life to having life, right? Like that's in front of us, that's near term, that's within the next 3 years we can build something like this. So, yeah, I think there's a lot that goes into that. It's not just us. It's not just the designer. You need like regulatory reform. You need a lot of other aspects. But there's a lot of good, like you mentioned, the story you're talking about. If you want to Google baby KJ, there's a lot of massive movement that's happening across the U.S., especially the regulation that's moving favorably.
So. And what does like multimodality look like in your space? Are there any analogies? How Sora is like video plus audio, like what is protein sequence prediction with
I think there's different ways of thinking of multimodality. Sometimes people think of you have proteins or you may have nucleic acids or small molecule ligands, but if you have like an all atomistic kind of approach toward it, it might all be represented the same way. Another way of thinking about it is you have protein sequence, you have structure, you have function, how do you incorporate all of that information. There's a lot of analogies with respect to multiple nodes. I think it's important. I think at the very least, that's going to be incredibly important for efficient learning from a pre-training perspective, and there's a lot of like controllable generation aspects to why multimodality can be important. So,
But do we think there's the intuition that multimodality will actually help capabilities in biology? Like it does in, I don't know, for self-driving with image, video, text, etc.
I think so. Yeah, absolutely. At least from an efficient learning perspective, I think it definitely will help.
But so then, in that case, why are you just a protein engineering company?
Oh, well, we've now moved beyond that as well. Yeah, that's a good question. So, for example, for gene editing applications, proteins don't operate in a vacuum.
Yep.
They interact with other proteins. They interact with DNA. They interact with RNA,
Yep.
Small molecules, ions, so on and so forth. And really, if you care about capturing protein function, right, the sequence to function relationship at the end of the day, you need to bake in all of that context. That's what we move toward. Yeah, absolutely. But the central element for us still is the protein, right? Like that's the executor of change. So we focus on that, but not with a blind spot, so to speak.
Intuitively, it makes more sense to have started with proteins as opposed to genomic language models like EBO, etc., or these small molecules. If eventually you're going to like use that as a scaffold to build other modalities.
Yeah, I think so. That's the bet we've made. Again, it's a combination for small molecules. It's a combination of like the reasoning there is there's a corpus of data that we can utilize. So that's the reason why we started there first. As opposed to EVO, I think EVO, so these are models that are like genomic foundation models, so just operating on DNA itself. I think that's a good research area. In terms of, like for us, like we want to draw the right balance where our research is grounded in the use cases, really at the end of the day, right?
It's commercially useful.
Yeah, it's commercially useful, societally useful, right? And I think if you have that lens, like research can be paralyzing in some sense because there's so much you can work on, right, in so many different directions. But in some ways, it's like freeing and comforting whenever you have like constraints in some sense, right? So that constraint like led us to more protein language models and stuff.
So maybe to wrap up, and if anybody has some questions, feel free to line up. But given that we're in this like trough of a bit disillusionment, I think we're all tired of hearing about this in biotech, but it is what it is. What do you think we're going to have to show? Like, what is the sort of like moonshot? I know you like to think about this regularly. Like the next major thing we can show that will get people like reinvigorated as to why we should be allocating effort and capital and be optimistic about the future of biology and biopharma.
Yeah, I think it's what I said before. Like, show a really amazing use case where there was no solution.
Okay.
You can't search within nature. You get the best biotech, scientists, for example, and they just can't come up with a solution, but AI can, right? I think that's the way forward, right? Like that's how you show real utility. That's how you kind of, pessimists are always writing the short term, but the optimists are writing the long term, right? Like that's how you really, drive home and kind of nail it in. So.